pictures, and I can. That's awesome. Okay, so yeah, this is a blood vessel. And let me back out. Okay. So I'm going to go all the way back to the lowest magnification and just give you an overview of the two parts of the pituitary. So this big, very bright pinkish purple area that I'm outlining is what is called the adenohypophysis, or anterior pituitary. And then this lighter version back here, this is the posterior pituitary, or neurohypophysis. So these two look super different because the neurohypophysis up here is nervous tissue. It's derived embryonically from the brain. And the adenohypophysis, or anterior pituitary, is derived from epithelial tissue. So when you are an embryo, you had a little pharynx, which is the back of your nose and throat, and a little pouch of that stuff formed and began to reach up toward the brain. Meanwhile, down from the brain, down from the diencephalon specifically, a stalk formed, and at the end of that stalk was a knob, and that stalk and knob reached down towards the epithelium coming up from the pharynx until the two joined and became enclosed in the sella turcica of the sphenoid bone. So the reason that these two tissues look different, the pars nervosa of the posterior pituitary. Pars nervosa means nervous system part, so that's this whole big blob. Looks light pink because it's primarily neurosecretory axons, so ends of neurons that secrete hormones, and then glial cells called pituocytes, which support the neurosecretory axons. Down here, these are epithelial-derived cells, so they look more epithelial or glandular in nature. And these guys are going to secrete a lot of different hormones. We're going to go through which particular hormones more so in lecture than in lab, but I am going to show you features that are on your lab module and which you do need to know. So let me begin by pointing out some features. One is, you'll notice if you're looking at your lab module right now that your lab module says be able to identify the pars distalis and pars intermedia of the adenohypophysis. So the pars distalis is most of what you see here, so I'm outlining it. The reason I'm taking care to move the tip of my pointer here instead of, for example, here, is because this middle part right here that I'm dragging the cursor through, this is the pars intermedia. You can actually, if you look closely, see the boundary between the pars distalis and pars intermedia. So see where my pointer is running? There's a faint line. So down here, it's a lot more reddish, and up here, there's a lot less red, and there's also these little fluid-filled pockets or spaces. And those are called Rathke's cysts. So that's one way to distinguish the pars intermedia from the pars distalis. So let's zoom in a little bit further here and have a look. So as you can see, there's a Rathke cyst, there's a Rathke cyst, here's another one, here's another one. And you can also see a lot more white space in here in the pars intermedia than you can down here. Now we're firmly in pars distalis territory. And you're going to see a lot of red in the pars distalis. That is because the pars distalis has a lot of sinusoidal blood vessels. So here's one right here, for example, and here's another, and another, and another, and another. You get the idea. So if you zoom back out, you can see pretty clearly that the sinusoidal blood vessels are really limited to the pars distalis. It's not the case that the pars intermedia doesn't have blood vessels, it just doesn't have nearly as many of them, so it looks paler in comparison, but not as pale as the pars nervosa. The pars nervosa is the bulbous portion of the posterior pituitary, which is the only part of it that you're seeing here. Another way to identify the pars intermedia is just to find the pars nervosa and find the pars distalis and then go right between them. The pars intermedia means intermediate part. It's literally between the two other parts. So that's also a good way to find that structure. We do not have the infundibulum on this particular slide because this is a transverse section, not a sagittal section. So the infundibulum is the stalk that connects the posterior pituitary, or neurohypophysis, to the hypothalamus. Okay, so there are a bunch of different cells in the pars distalis. You, fortunately, do not have to know them. So I'm going to show you some as an example, but you're not responsible for identifying them. And that's nice because there is a lot. So in the pars distalis, the reason it looks so colorful, also if you look up at this inset up here, where I'm hovering the cursor, you can see where we are on this slide, and I've zoomed in on an especially colorful region. So the reason that the pars distalis looks so much more vibrant compared with the pars intermedia or pars nervosa are primarily because of what kinds of cells are there. So these really darkly staining ones are basophils. So they like to stain dark and purplish in a hematoxylin and eosin stain. There are also acetophils, which are these really, really bright pinkish orange guys. And then we've also got chromophobes. Chromophobes means color fearing. So these are cells that don't like to pick up stain very much. So there's a chromophobe. There's another one right there. So any cell that's not dark purple or bright pink, basically. So the reason that these cells are different colors is because of the different hormones they produce. Again, you don't have to put a hormone to a cell type here. I'm just proving to you why the two regions of the pituitary are different. Okay, so let's zoom out a bit. And I'm going to use my cursor, and I'm actually going to bring back this little sidebar. So I showed you the acetophils and basophils and chromophobes. I showed you the capillaries. And then another nifty thing is that if you click on these little blue hyperlinks here, it'll navigate you to the area of the slide that is associated with the term. Unfortunately, it doesn't show you exactly what. So as you can see, I just clicked on, let me zoom out again and show you. So I'm going to click on pars intermedia. Boop. And it zoomed me in, but if I use my cursor here, can I have my cursor back? Where'd it go? Oh, cursor, where are you? That's strange. Oh, I think I have to drag it. There, got it. Okay. So if I bring my cursor back here, the hyperlink navigated me to here, but as you can see, there's still pars nervosa in the field of view, as well as a little bit of pars distalis. So the hyperlinks are helpful, but only if you already know what you're looking for. So what that means is the best way to approach this is that you're going to need to have your histology atlas open and look at the indicated or pointed at regions while you explore the slide yourself. And that's how you can best use Histology Guide. So I think that's just about it for the hypophysis. So let's go move on to a different gland. Hello. There we go. Okay, so I'm going to navigate down to the thyroid gland. And actually, I'm going to pick, you have some options here. So I like to go to the thyroid and parathyroid slide because you can see both of them together. This one also includes the thymus, as you can see. You're not responsible for learning the histology of the thymus, but it is there, so I'm going to point out what it is and how you can avoid it. The reason for that is that your lab slides, if you were to take 242 in person, also sometimes have hunks of thymus on them. So I'm going to show it to you just so that you can know that that's not what you're looking for. Blah, blah, blah. That's not that unusual, actually. So we have a bunch of slides that are like that. So let us collapse this. And then I'm going to wait for this to figure itself out. There we go. Okay, so I'm going to pick this. Let me grab my virtual pointer here. There it is. So I'm going to navigate to, I think, let's see, this top one. Yeah, I like that. Okay, so always a good idea to start out by looking at a low magnification to get your bearings. So I've got parathyroid here. The pink circles are thyroid, And then this really dark stuff is thymus. So this is what we're going to avoid because this isn't something that you're responsible for. You want this and this. And I'm actually going to zoom in on an area that is at the interface between the two so that you can compare and contrast them. Okay, so thyroid tissue is pictured here at the left where I'm hovering the pointer. And what you see are thyroid follicles. So each of these circular things is a thyroid follicle. And thyroid follicles are basically balls of simple cuboidal epithelium that are hollow. So imagine a simple cuboidal epithelium sphere that is a hollow ball. In the center of the hollow ball is stuff called colloid. So the definition of colloid chemically is a solid suspended in a liquid, typically a protein solid suspended in a liquid. But in this case, thyroid hormones are amino acid derived, but they're also carried on thyroglobulin. So there's a mixture of those things in here. So the follicular cells, and I'm going to navigate to a different follicle here, maybe this one. So only the cells like this one and this one and this one and these guys, only the cells that are directly bordering the colloid are follicular cells. These are the ones that are actually producing the colloid inside. And if you look closely, you can see these little sort of clear areas, right? These are areas where at the time of the tissue sampling or collection, the follicle was active. So these cells on the edge here are actively dumping stuff into the lumen of the thyroid follicle, which is why you see this little sort of cleared off space here. So this was an active thyroid at the time this tissue was collected. Okay, so let me zoom out a little bit, and I'm going to go try and find, let's see what Histology Guide has to say. Yeah, I like those. So the other kind of cell that you're going to look for are called C cells or parafollicular cells. So parafollicular means near follicles. C cells means clear because they don't like to pick up stain. So see how these cells here, these follicular cells, these are lining a follicle. These cells are not lining any follicle. They're just on the outside, and also they're not picking up stain very well. These are C cells or parafollicular cells. So the reason I'm emphasizing the difference is because follicular cells produce T3 and T4. So you do have to know hormones that these cells produce. Parafollicular cells or C cells produce calcitonin. So this is a hormone that you should be familiar with because calcitonin acts on bone. We discussed it in the skeletal system chapter. And these cells secrete thyroid hormones. So thyroid hormone, calcitonin. Thyroid hormone, calcitonin. These are C cells or parafollicular cells. These are follicular cells. Okay, so cell difference, hormone difference. Okay, so let's zoom out a bit, and I'm going to navigate over to my area where the parathyroid is. And let's zoom in on the sort of interface between the two of them. So down here we see very obvious thyroid glands, so follicles, follicular cells. I don't see any especially awesome C cells. There's a couple right here, for example. And then up here we have very different tissue. This is the parathyroid gland. So parathyroid glands, in most people there are 4 of them, and in most people there are 4 of them glued to the back of the thyroid. So that's why this is embedded in this tissue. They're a package deal. So the parathyroid gland consists of a very dense aggregate of cells. So you can see a lot of nuclei, and these cells are really closely packed together. And these cells are all going to produce parathyroid hormone. Parathyroid hormone should also be familiar to you because it acts on the bones as well, but it does the opposite thing of calcitonin. More about that will be discussed in lecture. So, parathyroid hormone. Hooray! Okay, so that's how to differentiate between the thyroid and parathyroid. Keep in mind, other things you're going to see in this tissue are adipose tissue and some dense irregular connective tissue and blood vessels. So just be aware, be a good practicing histologist, that just because a slide has a label that says thyroid gland does not mean that you won't find other tissues on that slide. Okay, let's go down to the adrenal gland. So the adrenal glands are located on the top of each of your kidneys, and they look like a little hat. This is a section of them. They're often triangular, but not in section, obviously. And you can see two very distinct regions. I'll zoom in a little bit. So you can see this very obvious middle region. It has big holes in it, and the cells are different colors than here. This is the adrenal medulla. So the adrenal medulla produces catecholamines, so epinephrine and norepinephrine. From here, where the pointer tip is, to here is the adrenal cortex. So the cortex produces many different hormones depending on which zone you're in. So let's examine the zones. And I really think that identifying these zones of the adrenal cortex is perhaps the most challenging piece of adrenal histology. So I'm going to spend most of my time talking about that. Let me see where they like for zona glomerulosa. Yeah, that's pretty okay. Okay, so the very, very edge, you can see that here's the connective tissue capsule. So we've got a dense, irregular connective tissue covering the outside of the gland, and you can see that there's blood vessels embedded in that connective tissue. And then we've got these little clumps of cells towards the edge here. So there's one, there's one, there's one, there's one, there's one, there's one, there's one. So it's the outer zone. It's only about, if you look at the sidebar, 15%. So it's pretty narrow. And the zona glomerulosa produces mineralocorticoids like aldosterone. So these are going to be a group of hormones that regulate water balance, for example. So aldosterone acts on the kidneys, and other ions, hence mineralocorticoids. So I'm moving the cursor a bunch here to point out the clumps of cells, also called glomeruli, in the zona glomerulosa. So roundish clumps of cells are a hallmark for zona glomerulosa, as is the fact that it's right next to the very edge of the gland and therefore right next to the connective tissue capsule. Moving down, we get to an area where the cells are arranged in columns. So I'm trying to find a place that I like. Let's see what they think about. Yeah, that's about where I thought too. Okay, so I'm going to zoom out a little bit. So the outer 15% is zona glomerulosa. So this is over here. If you move towards the medulla, you'll see that you stop having wads of cells and you start having these columns. So here's a column, here's a column, here's a nice column, here's another one, here's another one, here's another one, here's another one. So these are fascicles. A fascicle is a column or stack of cells. And the zona fasciculata is named after those fascicles. So this is a really nice one, so is this. And if we zoom really far in, I'm just going to give the screen some time to buffer. You'll notice that the cytoplasm of the cells of the zona fasciculata appears foamy. So you can see pink parts with interruptions, and they're like white bubbles, basically. The foamy appearance of these cells is because the hormones produced by the zona fasciculata are steroid hormones. So they're lipid-based, and they don't mix with water. Cytoplasm is water. So these like droplets of steroid hormone lipids suspended in the cytoplasm give the cells these foamy appearance, and they also arrange themselves into fascicles. So stress hormones like cortisol and corticosterone are produced here, and I'll show you in lecture the chart by which those are produced from cholesterol. All of these hormones coming from the zona fasciculata are derived from cholesterol. And then if you move further, you can see I'm tracing my pointer along a boundary. So to the left and above is the zona fasciculata, and then to the right and below, you start to see these cells that have less foamy and more pinkish cytoplasm. And they're also not really arranged into fascicles that much, so you get a change in color and a change in structural arrangement between the left and the right halves of the picture or the slide. So this area is the zona reticularis. And the zona reticularis makes estrogens and androgens. That's right. So this is like the meat of the zona reticularis down here. This is where it's really, really seriously reticularis-y. Another thing you'll notice about the zona reticularis, in addition to there's no more fascicles and the cells are much darker, is that there's more sinusoidal capillaries in the zona reticularis. So you tend to see them more here. So it gives the whole entire area a much darker, pinker appearance. So the zona reticularis produces sex steroid hormones, primarily androgens. Yes, that's right. Androgens like testosterone, for example. So we think of the sex steroid hormones as only coming from the gonads, when in fact they actually also come from the adrenal gland. And we'll talk more about what that means and what goes wrong with that in lecture. So, zooming out a little bit, let's go over our cortical zones of the adrenal gland one more time. Zona glomerulosa, zona fasciculata, and then right here, this dark pink area, no more fascicles, very dark cells, we've got zona reticularis. It's pretty easy to tell the difference between the reticularis and the adrenal medulla. So the cells in the adrenal medulla produce epinephrine and norepinephrine, and they're a lot purpler, so more basophilic. You also see very big blood vessels in the adrenal medulla. And that is because these cells in here are anaxonic neurons, so they're neurons with no axon, and their only job is to produce catecholamines, so epinephrine and norepinephrine. So these are going to take dietary tyrosine, convert those amino acids into catecholamines, and then dump them into the bloodstream. This helps with your fight or flight response, which by now you know all about from our discussions of the autonomic nervous system in 241. So this is where those adrenal medullary hormones, the catecholamines, come from and how they get into the blood. It's actually really easy to see. Okay, finally, we're going to go back to endocrine glands, and we're going to finish up by talking about the pancreas. So, here we've got the pancreas. So the pancreas is what is called a heterocrine gland, meaning that it's a mixture of endocrine and exocrine. So it's both. So what we're going to do is look at both parts. So I'm going to start by getting my cursor ready, and then we're going to get right to zoom in. So at low magnification, what you're going to see are a bunch of large ducts, as well as blood vessels. The tissue is pretty classically glandular. It's divided into lobules. So here we see one section divided from its neighbor by some connective tissue and blood vessels, and then another section. And then septa reach down into the exocrine tissue, forming lobules in here. Most of this very dark purple tissue is exocrine. So let's zoom way in. And I'm going to zoom in and... Alrighty. So, you can see in this section we've got little clumps of cells that are purplish or darkish purple-pink, and they're arranged into small balls or clumps. So here's one, here's one, here's one, here's one. These are called exocrine acini. That's A-C-I-N-I. Singular is an exocrine acinus, A-C-I-N-U-S. So a secretory acinus is just a clump of cells that excrete something. That's all. So the exocrine acini of the pancreas are going to excrete pancreatic juices. So this is a bicarbonate-rich juice with enzymes in it that helps you digest your food. Let's see, what else can we see in this view? We have blood vessels. So blood vessel, blood vessel, blood vessel. And if I zoom out a little bit, let me go find a duct. Oh, those are quite large. I'm going to find a small one first and then show you a large one. There we go. Okay. So here I have a duct. It's got a nice thick muscular wall, so this is made out of smooth muscle. This is going to help propel that secretion along. And then in the center here, I've got pancreatic juice. And then how do I know it's not a blood vessel? Well, A, this does not look like red blood cells. It looks like some kind of colloid. And B, it doesn't have simple squamous epithelium lining it. It's got simple cuboidal, which is a classic epithelial type of ducts. So that's how I know. All right, so that's one small duct. Let's look at a big duct. Where were you? There it is. Alrighty, so big duct. We've got simple cuboidal epithelium, lots of smooth muscle with some connective tissues. Here's an ancillary duct. Now, I realize in the lumen here, there's some red blood cells, which means that you might be confused and saying like, well, I thought you said that there weren't any red blood cells in the lumen of ducts. Keep in mind, in order for this tissue to be prepared this way, it had to be sliced. So sometimes red blood cells get to places where they aren't supposed to, like here. So what is your indication that it's not a blood vessel then? You have to look at the epithelium lining the lumen. If it is not squamous epithelium, this is not a blood vessel. Full stop. So be a good histologist, pay attention to your epithelial types, and you should be able to stay on top of whether you're looking at a duct or an artery, for example. Okay, so that was all exocrine stuff. The hard thing is to find the endocrine portions. And for this you have to use, remember those magic eye pictures where you stare at them and move them back and forth and cross your eyes, and then suddenly what was once a bunch of sort of random patterns becomes a unicorn or something? The pancreas is kind of similar. So relax your eyes, don't strain or try to focus. What you're going to notice is in the exocrine acini there are light patches. So there's one here, one here, one here, one here, one here, one here, and one here. Each of those is what's called an islet of Langerhans, or pancreatic islet. So here's one here. They're a ball of cells. They are lighter than the exocrine acini. So here is an exocrine acinus right next to some cells of a pancreatic islet. And look, the cytoplasm of the islet cells is pale pink. The cytoplasm of the acinar cells is dark purple. So there's a very distinct difference here. So islets of Langerhans produce two hormones that are antagonists of each other. They produce insulin, which encourages tissues to take glucose out of the blood, so it moves glucose from the blood into the tissues, and glucagon, which is the opposite. So glucagon helps to free up stored glucose and put it into the blood. Both of those hormones are secreted by this structure, the pancreatic islet. So let's go find some other islets here. And if you zoom way out, you can actually see they're all over the place. So there's one, 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 there's one. Here's a really nice one down here. You get the idea. So you're going to want to work on being able to find the pancreatic islets all by yourself. Okay, and that concludes the endocrine glands for this section of the material in histology. I hope you enjoyed the presentation, and I will see you next time.